A basic challenge confronts all viral therapies that most viruses have just a handful of genes, and they rely on proteins in the cells they infect, host cells, to perform many of the functions needed to reproduce. But therapies that target host cell proteins run the risk of killing uninfected cells, making matters worse. So antiviral therapies usually target something unique about the virus, something important enough that a few mutations in the virus won't make the therapy ineffective. Genes without DNA. Those of you who didn't sleep through high school biology may remember that genetic information is carried by DNA. When a protein needs to be built, the relevant bit of DNA is read and the cell makes a temporary copy of the information using a very similar chemical called RNA. This piece of RNA is then translated into a sequence of amino acids, which form the protein. While there are some exceptions to this, many RNAs perform important functions without ever being translated into proteins. All RNA in our cells is made by transcribing a DNA sequence. Our immune system is at the center of this. Whether you're browsing through social media, sitting in a cafe or walking through town, everyone is discussing the virus. It has sent the world into a state of chaos. To keep yourself and your loved ones safe, the best action is first to keep up to date and informed. Staying calm and aware, and preparing your immune system. All of us have an amazing protective mechanism called our immune system. It is designed to defend you against millions of bacteria, microbes, toxins, parasites and viruses. Viruses require a host, so they inject themselves inside your cells to thrive and replicate. As the virus multiplies, your immune system attempts to find the viruses and the cells infected and kill them before they can make more viruses. Often this fight can result in a compromised immune system, or when we already have a prior compromised system, our immune cells struggle to gain control. This is why the elderly population and those with a compromised immune system are particularly vulnerable to any viral disease. Ozone O3, a gas discovered in the mid-19th century, is a molecule consisting of 3 atoms of oxygen in a dynamically unstable structure due to the presence of mesomeric states. The gas is colorless, acrid in odor and explosive in liquid or solid form. It has a half-life of 40 minutes at 20 degrees Celsius and about 140 minutes at 0 degrees Celsius. Its basic function is to protect humans from harmful effects of UV radiation. Ozone occurs at less than 20 grams per cubic meter from the Earth's surface at concentrations that are perfectly compatible with life. Although O3 has dangerous effects, yet researchers believe it has many therapeutic effects. The beginning of precise medical O3 generators has only recently allowed the mechanisms, action and possible toxicity of O3 to be evaluated by clinical trials. Ozone has a capacity to oxidize organic compounds and has well-known toxic effects on the respiratory tract when present in smog. In medical use, the gas produced from medical-grade oxygen is administered in precise therapeutic doses, and never via inhalation, and advocates that it has excellent health benefits in dental caries, decreased blood cholesterol and stimulation of antioxidative responses, modifies oxygenation in resting muscle and is used in complementary treatment of hypoxic and ischemic syndromes. Ozone therapy is a highly effective treatment and prevention for any type of viral infection. Why you need ozone to prevent the virus? Ozone plays a fundamental role in the treatment of viral infections and has proven to be effective for previous infections such as SARS, severe acute respiratory syndrome, and MERS, Middle East Respiratory Syndrome. Ozone therapy is effective due to its direct and indirect mechanism of action. Ozone therapy activates the immune system. Anti-microbial, antibacterial, antifungal and most importantly its antiviral effects. Ozone therapy decreases inflammation. Ozone is a powerful mitochondrial stimulant. Ozone leads to an increase in the amount of oxygen released to the tissues, resulting in improved circulation. Ozone therapy activates NRF2 pathway, is the master regulator of the cellular antioxidant response. Ozone therapy causes an increase in the red blood cell glycolysis rate. Ozone increases our most potent radical scavenger and cell wall protectors, glutathione peroxidase, catalase and superoxide dismutase. To prevent and protect the body from a viral infection, with ozone it is administered via rectal insufflations, ozone water and sinus insufflations. This is the easiest, most inexpensive, safe and effective protective solution that everyone should have available to them, their family and loved ones. Ozone exploits the redox vulnerability of deadly viruses. Ozone's natural unique physical, chemical and biological properties towards boosting our immune system, destroying viral and other microbial infections, increasing our antioxidant pathways among many others make it the most versatile therapy available. History of ozone therapy. Ozone therapy has been utilized and extensively studied for many decades altogether. Its effects are proven, consistent and with minimal side effects. Medical O3, used to disinfect and treat disease, has been around for over 150 years. Used to treat infections, wounds and multiple diseases, O3's effectiveness has been well documented. It has been used to disinfect drinking water before the turn of the last century. Ozone was known to treat as many as 114 diseases. Ozone therapy has been in use since the 1800s, and in 1896 the genius Nikola Tesla patented the first O3 generator in the US, later forming the Tesla Ozone Company. During the First World War 1914–18, doctors familiar with O3's antibacterial properties, and with few other medical resources available to them applied it topically to infected wounds and discovered O3 not only remedied infection, but also had hemodynamic and anti-inflammatory properties. In the late 1980s, reports had emerged that German physicians were successfully treating HIV patients with O3 AHT autohemotherapy. There was then no pharmaceutical treatment for HIV and a pandemic was feared, so Canadian authorities authorized the study to test safety and efficacy of O3 AHT in AIDS patients. Ozone had shown promise in vitro testing. Ozone was seen effective at disinfecting extracorporeal blood samples of HIV. Unfortunately for AIDS patients, O3 AHT proved to be an in vivo ineffective treatment. SARS and ozone. Ozone is a naturally occurring energy-rich molecule embodying unique physiochemical and biological properties suggesting a possible role in the therapy of SARS, either as a monotherapy or, more realistically, as an adjunct to standard treatment regimens. Owing to the excess energy contained within the O3 molecule, it is theoretically likely that O3, unlike organism-specific antiviral options available today, will show effectiveness across the entire genotype and subtype spectrum of SARS. Mechanism of action: inactivation of bacteria, viruses, fungi, yeast and protozoa. Ozone therapy disrupts the integrity of the bacterial cell envelope through oxidation of the phospholipids and lipoproteins. In fungi, O3 inhibits cell growth at certain stages. With viruses, the O3 damages the viral capsid and upsets the reproductive cycle by disrupting the virus-to-cell contact with peroxidation. The weak enzyme coatings on cells which make them vulnerable to invasion by viruses make them susceptible to oxidation and elimination from the body, which then replaces them with healthy cells. Stimulation of oxygen metabolism. Ozone therapy causes an increase in the red blood cell glycolysis rate. This leads to the stimulation of 2,3-diphosphoglycerate, which leads to an increase in the amount of oxygen released to the tissues. Ozone activates the Krebs cycle by enhancing oxidative carboxylation of pyruvate, stimulating production of ATP. It also causes a significant reduction in NADH and helps to oxidize cytochrome C. There is a stimulation of production of enzymes which act as free radical scavengers and cell wall protectors, glutathione peroxidase, catalase and superoxide dismutase. Production of prostacylin, a vasodilator, is also induced by O3. Activation of the immune system. Ozone administered at a concentration of between 30 and 55 grams per cc causes the greatest increase in the production of interferon and the greatest output of tumor necrosis factor and interleukin-2. The production of interleukin-2 launches an entire cascade of subsequent immunological reactions. Mechanism of action of O3 on the human lung. Ozone exposure induces a significant mean decrement in vital capacity. It significantly increases mean airway resistance and specific airway resistance but does not change dynamic or static pulmonary compliance or viscous or elastic work. It also significantly reduces maximal transpulmonary pressure. And furthermore significantly increases respiratory rate and decreased tidal volume. How does ozone destroy viruses? Viruses are very simple microbes, they can't do much by themselves, and instead they need a host cell. Viruses get inside a host cell and take over the controls. Once they have taken over, they very quickly replicate more viruses and releases them into the body. Ozone destroys viruses by diffusing through the protein coat into the nucleic acid core, resulting in damage of the viral RNA. Numerous families of viruses including poliovirus IN2, human rotavirus, Norwalk virus, parvoviruses, hepatitis A, B, adenoviridae, picornaviridae, poliovirus, Coxsackie, echovirus, rhinovirus among many others, are susceptible to the virucidal actions of ozone. Unlike healthy cells, which possess complex enzyme systems, superoxide dismutase, catalase, peroxidase, virus have no protections against oxidative stress, therefore making virus vulnerable to ozone. Repeated oxidative stress caused by ozone causes the production of NRF2 protein, which is able to initiate the production of proteins favoring most of the cell functions. What is NRF2? NRF2 is a powerful protein that is latent in every cell. It is a switch for 200 or more different genes, all of which code for different aspects of cellular defense such as immune and inflammatory responses, tissue remodeling, fibrosis, carcinogenesis, metastasis, and even cognitive dysfunction and addictive behavior. NRF2 is unable to operate until a potent NRF2 activator like ozone releases it. Once released, it migrates into the cell nucleus and bonds to the DNA at the location of the human antioxidant response element HAIR, which is the master regulator of the total antioxidant system that is available in all human cells. When NRF2 is activated in the nucleus, it turns on the production of antioxidant enzymes such as catalase, glutathione peroxidase and superoxide dismutase (SOD) are most powerful neutralizers of free radicals that our body produces. Biological responses induced via the activation of NRF2 are, increasing the levels of antioxidants, stimulating GSH regeneration via glutathione and thioredoxin reductase, increasing the levels of enzymes that detoxify oxidants and electrophils, catalase, SOD, GPX, NQO1, H01. Increasing the levels of phase II detox enzymes inhibiting cytokine-mediated inflammation, reducing iron overload and subsequent oxidative stress, recognizing, repairing and removing damaged proteins, protecting from apoptosis induced via oxidative stress, increasing DNA repair activity. To put it simply, the NRF2-dependent antioxidant response has been shown to protect against oxidative stress-related diseases such as cancer, neurodegenerative diseases, Alzheimer, Parkinson, ALS, Huntington's disease, cardiovascular disease, infections, lung emphysema, inflammation and aging. Function of ozone therapy. Ozone therapy activates NRF2 pathway. Ozone increases the our most potent radical scavenger and cell wall protectors, glutathione peroxidase, catalase and superoxide dismutase. Ozone therapy disrupts the integrity of the bacterial cell envelope through oxidation of the phospholipids and lipoproteins. In fungi, O3 inhibits cell growth at certain stages. With viruses, the O3 damages the viral capsid and upsets the reproductive cycle by disrupting the virus-to-cell contact with peroxidation. Ozone therapy causes an increase in the red blood cell glycolysis rate. Ozone therapy leads to an increase in the amount of oxygen released to the tissues. Ozone activates the Krebs cycle by enhancing oxidative carboxylation of pyruvate, stimulating production of ATP energy. Ozone therapy increases the production of interferon and the greatest output of tumor necrosis factor and interleukin-2. The production of interleukin-2 launches an entire cascade of subsequent immunological reactions. Activation of the immune system. Ozone therapy stimulates the neuroendocrine system and therefore produces a feeling of well-being. Ozone therapy combats aging. Ozone therapy decreases inflammation through the activation of NRF2 and subsequently the suppression of NF-κB, pro-inflammatory signaling pathway. Millions of ozone treatments have been performed over the last 4 decades. Therefore, ozone therapy is considered very safe, very cheap and an easy treatment that can be done as a best alternative therapy which can be used instead of conventional medical treatment in the history of medical science.